Great. Thank you for the introduction, and looking forward to the opportunity to present some of the research that we're working on, looking at these underlying biological properties of CAR T cells that favor tumor cell killing, and of course, using the Lightning single cell platform. And so just to give a, general introduction and kind of an overview of where we're at in the field. So, cancer immunotherapy is now established as a major therapeutic modality, with both immune checkpoint blockade and adoptive cell therapy for various types of cancer. Solid tumors, and particularly those with high tumor mutational burden, have shown to respond reasonably well to immune checkpoint blockade, which provides evidence that the immune system, and particularly CD8 T cells, are capable of resolving human tumors. And certain hematological malignancies have benefited from the advancements in cellular therapeutic approaches. We see this primarily with the advancements in different CAR T cell therapies. But despite these successes, there are still major limitations to these approaches and ongoing questions that need to be addressed. And so when we're thinking about the factors that, contribute to an effective anti-tumor response, and, thinking about the different parameters that we want to consider when developing cell-based therapies, you can kind of distill this down to a very simplistic view by looking at those that are tumor-centric. And so these would be, the type of targets that you're selecting to target by your therapeutic approach. Tumor antigens, you want these tumor antigens to be different than self-antigens in order to prevent off-tumor toxicity. And then you want these to be necessary for tumor cell survival. So, these driver mutations that in the absence of these mutations, you wouldn't have effective, transformation and persistence of your tumor cells. And so in this illustration, basically what I'm highlighting is that if you're moving from right to left, where you're looking at, antigens that are present on both cancerous cells and healthy cells, in those scenarios, you're going to have an increased likelihood of having off-tumor toxicities. And as you move further down, from right to left, you have, single nucleotide variants, which can be point mutations that are going to be selectively present on the tumor cells and not present on healthy cells, or fusion mutations where you get two genes that fuse together that the immune system, would recognize as essentially as foreign and be able to eliminate. And so, of course, when we're thinking about developing these cell-based therapies, we want to focus on, targets that are selectively present on tumor cells and that are going to limit any off-tumor toxicity. Now, the other component is thinking about, the immune-centric parameters. So, we want to make sure that there's, sufficient immune cell infiltration. And so this is, commonly referred to as hot versus cold tumors. And if there isn't good immune infiltration, we want to discover or determine ways that we can increase the extent of infiltration into the tumors. We want a robust response, meaning that we target multiple antigens preferentially in order to prevent any likelihood of immune escape. And then lastly, when we're thinking about just the evolutionary process of the immune system, there's this high degree of self-tolerance, and you want a balance of specificity and tolerance, right? And so typically, these tumor neoantigens are going to be unique in that they're going to be, more distant than self and maybe more similar to, pathogenic mutations. And so those are ones that we would favorably target. Unfortunately, this isn't, always the case. There are, still shortcomings and lingering questions. In certain types of cancers, we can't, or we don't currently have targets that are well identified. And so, this has been a major gap in the field when we're thinking about, how we can identify these targets and which are going to have the highest likelihood of driving clinical benefit. And as I mentioned, a lot of the early studies really focus on adult solid tumors with high tumor mutational burden. There's been a, fairly narrow range of cancer neoantigens that have been explored. And then there's, really a lack of commercially available reagents to really thoroughly explore, the comprehensive immune response to these different cancer neoantigens. And then, we can utilize some of these prediction algorithms to predict which neoepitopes are going to be most immunogenic and potentially most favorably targeted when we're developing these cell therapies. And so this makes it difficult to extrapolate findings, right? We don't know whether, different tumor types are going to have, differential responses. And we don't know whether we can extrapolate between patients, and so sometimes it's difficult to think about, a one size shoe fits all approach where, one therapy is going to derive benefit across multiple patients. And then lastly, we have an incomplete understanding of the mechanisms that govern tumor immunogenicity. And so, we really need to further understand whether there are specific phenotypic or functional characterizations that can be correlated with clinical outcome. And so in our studies, one of the major bottlenecks is really in identifying, the tumor-specific T cells more from a validation and characterization standpoint. And so, we utilize a lot of different tools and technologies in our discovery phase where we can, perform whole genome or whole exome sequencing and identify, patient-specific mutations. But once we identify these, it's really cumbersome to develop different assays and techniques in order to validate that, these targets that we're suggesting are good targets are actually, inducing an immune response. And then furthermore, characterizing how the immune system is specifically recognizing these. And ultimately, the goal is to utilize this information to advance the translational capabilities of these therapies and to further develop cell-based therapies, which we can then use to treat patients. And so in this talk, I'm going to focus primarily on some of the work we're doing in CAR-T cells, but the same idea applies to different T-cell receptor T-cell based approaches and pretty much any cell-based therapy that you're utilizing to characterize and target cancer neoantigens. And so, in collaborative studies with Dr. Nirav Shah and Dr. Brian Johnson here at MCW, one area of our research focuses on trying to improve the in vivo persistence and clinical efficacy of dual CAR-T cells. So these are CAR-T cells that target anti-CD19 and CD20 by assessing the long-term outcomes in patients with non-Hodgkin lymphoma and leukemia. And really, there's various objectives, that we're looking to discover. Some of the experiments that we're performing currently are to test whether, different priming conditions during the manufacturing process. So as you're generating your CAR-T cells and you're utilizing different cytokines, during this priming, how can that impact the end product and how will that result in differences in the T cell functionality and clinical outcome? A major area of focus is actually screening for desired phenotypes and functional capabilities of specific CAR-T cells, within a heterogeneous population of cells. And so that's something that you're going to hear me kind of echo throughout my talk is, understanding the heterogeneity of the CAR-T cells that are responding differentially and then trying to identify those that are inducing the most favorable responses. And then lastly, determining whether mechanisms of therapeutic response correspond to specific features of CAR-T cell biology. So are there underlying, parameters in terms of, the T cell biology that outside of the manufacturing process that governs their ability to either be, polyfunctional in terms of their ability to produce multiple cytokines, or that influences their ability to perform their cytotoxic functions. And so, typically when we're performing these assays and trying to unravel the cellular complexity and the heterogeneity in the, cell-based immune therapeutic responses, we typically utilize cytokine secretion and cytotoxicity as two of our main readouts. Now, one of the limitations of many of the bulk assays is that they don't provide sufficient resolution to really understand the heterogeneity and then to, start to extrapolate whether or not, these differences in specific subsets of CAR-T cells are the ones that are mediating, clinical efficacy. And so these are just, some different assays that we typically use and, some limitations for each of these. And so, typically when we're looking at cytokine secretion, we'll utilize ELISAs or ELI spots or, intracellular cytokine staining. And so really these assays are great in being able to determine specific cytokines that are being produced, during cold culture experiments. But unfortunately, you don't get sufficient resolution to know which specific cells are responding during these assays. Similarly with the cytotoxicity assays, typically you're looking at bulk samples. A lot of these assays are terminal, meaning that you can't recover the cells after performing the assays. And a lot of them require a large amount of starting material, which can be a limitation depending on, the amount of cells that you have in your tumor biopsies, or the amount of, cells that you have available to perform these multiple assays. And so I'm not going to go through, each of these points in this table, in detail, but to really highlight is that, in the lab, when we're looking at determining, the different readouts, we kind of have to go through the pros and cons of each of these approaches and determine, which we want to perform. And unfortunately, due to limitations in starting material, oftentimes we're not able to perform multiple assays. And so we kind of have to prioritize depending on, our specific questions that we're looking to answer. And so, oftentimes we have to either prioritize looking at cytokine secretion versus cytotoxicity. And so, these are some of the limitations and some of the caveats to performing each of these assays. And so really, one of the main criteria for us in terms of thinking about, comprehensively assessing the CAR T cell response was trying to figure out a way that we could link all of these parameters utilizing, a smaller amount of starting material and then being able to perform these assays in unison. And so, this is where the Lightning platform really shines in that, you're able to look at multiple parameters simultaneously and longitudinally. And so just to provide a little bit of overview of the Lightning platform, so essentially I like to think of it as a microscope, a flow cytometer, and a microfluidic device all in one. So essentially you can, monitor in real time how your co-culture experiments are being conducted. So, it has a capability of looking in bright field and then looking at different fluorescence parameters to characterize, the size, the morphology, and specific functional characteristics of your cells of interest. Additionally, you can measure, the secretion of various analytes. And so, as I mentioned, one of the main parameters that we're looking at when determining which of our CAR T cells are most functional is cytokine secretion. And so you have the ability to look at multiple cytokines at the same time and at single cell resolution. Additionally, you can perform multiple assays on one chip. And so I'll go over a little bit more on the next slide what this looks like exactly. And you can look at these parameters longitudinally. And so you're really getting a robust picture of exactly how your CAR T cells are responding and the differential responses between different CAR T cell populations. Lastly, you can culture the cells on chip since this is an enclosed sterile environment. And then you can, export selected cells of interest for further downstream assays. And so, as I mentioned, this is a major limitation in a lot of the other assays that we typically utilize in the lab in terms of them being, terminal in nature and not being able to recover the cells after performing, specific assays of interest. And so just to go over, some of the characteristics and, kind of the one of the most interesting components of this Lightning platform, there's two main features. There's this optoelectric positioning and the optoselect chip. And so essentially what, this allows you to do is to perfuse your cells or your beads of interest onto this chip using, microfluidics. And then using this optoelectron positioning, which is a non-destructive light source that allows you to selectively pan, your cells at single cell resolution into these pens on the microchip. And so these chips allow you, to perform these assays over time and then to, characterize and collect multiple parameters of information, to look at, cytokine secretion, cellular cytotoxicity, proliferation, and multiple other assays on chip. And so to highlight, one of the main assays that we've been utilizing to characterize the polyfunctionality of different CAR T cells, we've been using the cytokine secretion assay. And so this assay allows you to customize which cytokines you want to capture by introducing specific capture beads into each of the nanopens individually. And then you can co-incubate, your T cells with the tumor targets and measure cytokine secretion in real time. And so I'll go, through each of these steps in subsequent slides, but just to kind of provide an overview of how this is done, in step A, you would add your capture bead of interest. So say you're interested in looking at interferon gamma, you can add your capture bead into each nanopen Subsequently, you could add your T cells into those same corresponding pens, and then add your target cells of interest. And so oftentimes we use, Raji cells or tumor cell targets in order to monitor how the CAR-T cells are responding to these tumor targets. And then you can, perfuse with media and in co-culture these single cells, anywhere from 24 to 48 hours. You can add additional surface markers, antibodies for additional surface markers. And then essentially you perform a capture ELISA where your capture beads are, binding to the secreted cytokines. And then you come in with a secondary antibody that binds to a different epitope on your cytokine. And then you can measure which cytokines are produced by your T cells in this co-culture assay. On the right, I just want to point out that you can utilize multiple cytokine capture beads. And so in a lot of our assays, we're looking at, IL-17A, interferon gamma, TNF alpha, or IL-2. And what we're able to do is, due to differences in the fluorescence in the Cy5 channel, we could distinguish between each of these different cytokines as they're being produced. Additionally, we can utilize size as a parameter. And so these beads have different sizes. And so you can distinguish, those that are selectively binding, IL-17A or interferon gamma. And so it gives you a great deal of flexibility to kind of set up your assay to, understand which cytokines are being produced on a single cell level. And so this is, the first step, as I mentioned, and just to give an overview of how this is happening on chip. And so these, let me go back. And so I'm not able to play the videos, unfortunately, but just to kind of highlight, so that, you use this these cages around your beads, and then they're going to selectively pen in the capture beads into each of these nanopens. Subsequently, you would load your T cells into those same pens that have the capture beads, and then you would add your target cells, and then add any of the surface antibodies in order to phenotype your T cells, and also to, as I mentioned, perform this assay similar to a capture ELISA, where you have a secondary antibody to another epitope on your cytokine of interest in order to determine which cytokines are being produced in each pen. And so another, characteristic and unique feature of this is that you can multiplex these different workflows or these different assays with, different assays such as cytotoxicity, or to look at transcriptional profile in these same pens. And so, after performing these assays and detecting which cytokines are being produced, you're also able to look at, how your T cells are specifically targeting your tumor cells or your target cells of interest. And so typically when we perform these assays, we'll label the target cells with either CFSE or GFP, and then we add a caspase-3 reagent, which is perfused during the imaging steps, and allows us to measure, as the T cells are killing our tumor targets. And again, you're able to do this longitudinally and monitor this process in real time. And so this is an example of some of the heterogeneity that we're able to unravel utilizing these assays. And so in the furthest left panel, what you can see is that we loaded a single CAR-T cell, a single target cell, and then our cytokine capture bead. Now, in the no killing, early killing, and late killing panels, these are all individual pens, where we are able to see that there's an immune synapse formed between our CAR-T cells and our tumor targets. But in some scenarios, as we're measuring these responses longitudinally, some of the CAR-T cells aren't killing their tumor targets. Others are killing very early, and then others are killing at a much later response. And so it really allows us to understand not only, the ratio of cytotoxic CAR-T cells, but also differences in the kinetics of this response. And as I mentioned, we're able to monitor this, in each pen. And so on the OptoSelect chip that's utilized on the Lightning, we can measure 1,500 pens simultaneously and then generate these histograms at the bottom to determine the kinetics of specific CAR-T cell killing. And so what we're able to decipher is whether or not, in our patient samples, do we have, higher frequency of non-cytotoxic CAR-T cells, of rapid cytotoxic CAR-T cells, those that are slowly cytotoxic, or those that are serial killers, meaning that if we continue to add tumor targets into the pens, are these CAR T cells able to continue to kill additional tumor targets, and how long and how many can these CAR T cells actually kill? This doesn't look like the slide is advancing. 1 second. Okay, here we go. And as I mentioned, really one of the main strengths of this platform is being able to look at multiple parameters simultaneously. And so what you can see here is that, under different priming conditions, we can compare both the cytokine secretion. So on the y-axis here, we're looking at interferon gamma MFI, and on the x-axis, we're looking at cytotoxicity in minutes. And we're able to determine, the relative abundance of CAR T cells under each of these priming conditions that we are characterizing as fast killing and high interferon gamma producers, those that kill much slowly or much more slowly and produce high levels of interferon gamma, those that kill quickly but produce low levels of interferon gamma, and those that are both slow killers and produce low levels of interferon gamma. So this really allows us to understand the complexity of how different CAR T cell populations are responding, and really start to unravel then the characteristics that are going to be most efficacious when determining, CAR T cell persistence and clinical efficacy. And so these pie graphs highlight, the differences in these priming conditions, and how when we, prime with IL-7 and IL-15, we end up generating CAR T cell products that are much more polyfunctional, and we have fewer that are non-functional, meaning that aren't producing cytokines or aren't being cytotoxic. And so this helps us, when we're deciding which different CAR T cell manufacturing procedures to utilize, and whether or not we'll see greater clinical efficacy when we utilize these CAR T cells in patients. And then lastly, we're able to look at each of these parameters on the same cell. And so as I mentioned earlier, one of the major limitations of some of the assays we traditionally utilize, in the lab, is that, we can look at each of these parameters in isolation, but to really understand, the cells that are going to give us the greatest likelihood of being, of persisting in vivo, and that are inducing, greater levels of antitumor cytotoxicity, we want to understand each of these parameters and be able to link all of them to the same cell. And so what you're seeing here is that we're looking at both IL-17 alpha MFI, interferon gamma, and then caspase MFI, but then also the size of the either circle or square allows us to look at how quickly they're killing. So those that have the larger circle or square means that they're killing much more quickly. And then we're able to, look at additional parameters. So looking at whether the CD4 or CD8 CAR T cell products are killing preferentially. And then we can, again, compare different priming conditions. So comparing IL-2 versus IL-7 and IL-15 prime CAR T cells. And so ultimately what the goal would be is for us to be able to, link all of these parameters and to then begin determining, which of these characteristics are going to correlate with, the greatest clinical efficacy. And so, for example, if we wanted to characterize the surface phenotype and cytokine secretion of specific CAR T cells, and then link that to their killing potential, we could really start to understand some of the underlying biology that's mediating, greater clinical efficacy and greater anti-tumor responses in vivo. And so with that, I want to acknowledge, the various members in my lab that have, helped, conduct these experiments. I also want to acknowledge Dr. Brian Johnson. He's the director of the Cellular Therapy and Immune Monitoring Core at MCW. And then Dr. Nirav Shah, he's a clinician that's actually performing a lot of the clinical trials. And he's actually giving a talk later this afternoon. So if there's any interest in understanding more about some of the clinical trials ongoing at MCW utilizing these CAR T cells, be sure to check out his talk. And then I also like to acknowledge the entire team at Berkeley Lights for, providing guidance and an overview of the different workflows that are available, and working with us to really get the system up and running at MCW so that we can, continue to advance cell-based therapies and provide, greater efficacy in the cell-based therapies that we're utilizing to treat patients with cancer. And so with that, I'm open to taking any questions.
Terrific. Thank you very much, Anthony. And we've got an absolute load of questions here. We're not going to be able to get through them all. So I'll start with some which may be suited to single word answers. How many cytokines can you probe on a single chip?
And so typically on a per pen basis, we typically look at up to 3. And so that gives you, the ability to utilize size and different, fluorescence of those capture beads. So typically, 3 is what we've been utilizing in our assays.
Terrific. And related to that, does it provide absolute quantitation of the cytokines or more of a relative MFI signal?
Yeah, so typically, you're getting more of a relative MFI, right? So you're essentially seeing whether or not the cytokine is being produced. There are ways for you to quantify it by utilizing MFI, but typically we're just looking at, which cytokines are being produced, are each of the T cells producing one or another or multiple cytokines.
Great. And what is the sensitivity of the cytokine assay?
So so I'm not entirely sure what the range is, but so the cytokine capture beads are similar ones that are used in cytometric bead arrays, and so I can't remember what the lower limit of detection is, but it is pretty robust, right? Because it's similar to what you would see in an ELISA, since it has the capture bead and the secondary antibody for another epitope on that cytokine. I don't know exactly what the lower range or lower limit of detection is, though, off the top of my head.
Okay. How do you see your observations being adopted into manufacturing processes? I think that's probably the most important question.
Sure. Yeah. So I think it would, it's going to provide a lot of insight in terms of, our specific manufacturing processes, like, the different priming conditions, whether or not you're using fresh or cryopreserved, and whether or not, they're going to be more polyfunctional, and you can actually look at the heterogeneity of the response. That's going to be one of the main influences in helping dictate how you would utilize, this platform to address those questions. So you could prioritize a specific manufacturing process based on, what you're seeing, in these assays.
Great. And another question here, what has been the best strategy to ensure that the target cell and CAR-T cell encounter one another?
That they encounter one another? Was that the question?
Yes, yeah, that's correct.
Okay. Yeah, so the chip is actually, has a slight incline, and so it allows, just due to gravity for the cells to come in contact, on the in each of the pens. But what we see in a lot of, if you monitor the bright field images, once that immune synapse is formed, you can actually get movement of your target cells and your T cells, and they follow each other around, or the T cells follow the target around in the pen. And so, so due to the size of the pens, there's a high likelihood that the targets and the T cells will come in contact. And as I mentioned, due to how the chip is positioned in the system, it allows them to, come in contact closer towards the bottom edge of those pens.
Great. Well, Anthony, I'm very sorry, but we've actually run out of time. I'm sorry for the people out there where we didn't manage to get to your questions, but don't forget there's the stage chat, and Anthony will be available there. Please go and ask your questions directly. So thank you very much indeed. It was a terrific presentation, Anthony.